Okay, good afternoon, everyone. My name is Ondrej Uhlík, and I come from the Department of Biochemistry and Microbiology. And my task here will be to introduce you to the project, one of the projects that we are dealing with in the Laboratory of Applied Microbial Ecology. Before I can do that, I should probably introduce what microbial ecology is. So, first of all, I will start with something that happened a long time ago, at the time of Robert Koch, who actually postulated a couple of important postulates that biology and biochemistry still lean on. And from his times, it was already pronounced that it is very important to use pure culture in microbiology. So ever since Robert Koch, microbiology was pretty much the science of pure culture. However, the problem that we have with the pure culture is that in the environment, microbes are seldom present on their own. They are usually present as complex communities, and these communities contain a lot of different taxa, a lot of different genera, a lot of different species, and these species together widely interact, and that's very important. You can see as an example here a picture from Yellowstone National Park, where you see different colors ranging from yellow to brown, and all of these colors are basically derived from different populations that are contained in these parts of the Grand Prismatic Spring. Another example can be given by Vinogradsky column, which is like a model, simple, small ecosystem that works just because it contains a lot of different kinds of populations, and each part of the community performs some activity, and this ecosystem can only be working because the populations in the ecosystem interact. So until now, I have used a couple of times the word interact. And that's what microbial ecology is about. It studies the interactions of microorganisms with their environment, with each other, with the plants, with animals, basically with all macroorganisms. And microbial ecology as a discipline studies microbes and their involvement in biogeochemical cycles, and also the interaction of microbes with some anthropogenic effects, and it's mostly about pollution or climate change. And why is it important to study or to deal with microbial ecology is that once we are capable of understanding some ecological mechanisms, we will be able to somehow, to a better extent, predict, conserve, and manage microbial communities to some desired outcomes. And by these desired outcomes, we can have a lot of things on our minds. First of all, we can answer some basic questions, including why are we here? Or we can have a little bit more practical questions, such as how can we improve our lives? And by that, I mean that we can use microbes to things such as environmental restoration, food production, production of antibiotics, and things like that. Also, studying these interactions among microbes can help us somehow measure the effect of climate change, land usage, and things like that. So, among these interactions that are very abundant in the environment, we have plant-microbe interactions. And plant-microbe interactions are also plentiful in the environment. I can mention just a couple of them because of the time. For instance, due to plant-microbe interactions, nitrogen fixation is possible, mycorrhiza is enabled, also plant is capable of being protected from pathogenic species, and so on. And this is all given by the interaction. And it looks like that in these interactions there is a couple of molecules that are very important for these interactions, and these are called secondary plant metabolites. Basically, it's a wide array of compounds, mostly terpenes, phenolics, and alkaloids, and they have probably evolved in the plants for multiple reasons. A couple of examples can include that these compounds can help the plant tolerate frost. They can help them store nutrients. They can act as signaling molecules. They can act as allelopathic molecules, or they can also help the plant protect from the herbivory. And when I turn this into the ecology of soil, we have a couple of facts about secondary plant metabolites, and based on these facts, we have a hypothesis-driven research. So what we know is that secondary plant metabolites are pretty much continuously released into the environment, into soil, through a couple of processes such as root exudation, root turnover, or litter decomposition. And once they enter the soil, they can have multiple functions. They can act as carbon sources to bacteria, energy sources. Sometimes they can have antimicrobial activity, or they have ability to disrupt communication between bacteria. And with all of this in mind, what we are doing in our lab is that we are testing a hypothesis that secondary plant metabolites are some of the key features that basically control soil microbial community structure and composition. So pretty much we are thinking that what is out there in the soil is basically governed by the composition and concentration of secondary metabolites in plants. So that's one of the hypotheses that we test in our laboratory. The second couple of facts, or second group, is that we know that due to changes in the environment and changes in selective pressure, the concentrations and contents of secondary metabolites in plant need to change. And therefore, bacteria had to develop enzymes with broad substrate specificity in order to degrade as many secondary metabolites as possible at once. And we use this in order to test our hypothesis that enzymes that originally developed for the detoxification of species are also involved in the degradation of anthropogenic pollutants, because these are somehow structurally similar to secondary metabolites. So thereby, we are claiming that secondary plant metabolites are pretty much covering the biodegradation potential in microbial communities. Wow, it was short. So thank you for your attention, and these are my colleagues from the lab who are helping with the research going on.